Thank you.
Thank you very, very much for being here. So, yes, so we wait a little bit until everybody has jumped. And I think that maybe we are now, yes, I think everybody is here. Great. So thank you, Doug, for being here all the way from Arizona, from the University of Arizona. So it's very early in the morning, but apparently you're an early riser. And so Doug is going to talk about ethics in imaging today, and I'm very excited about this lecture. So please, go ahead.
So one disclaimer, this background is actually a sunset, not a sunrise, you know, if we're going to do truth in imaging.
Oh, yes.
All right, let's see here. Are you seeing my screen now?
I think it's coming now. We see. Yep.
Okay. So I thought I'd show you some a little local color. But thank you so much for asking me to be here this morning or this afternoon in your case. Let's see here. So one of the things I noticed when I looked at last year's lecture was a lot of talk about ethics in general. And I just wanted to touch on the fact that when you're working with ethics, it's a broad topic. Certainly in the United States, human subjects research, we have a less than stellar history as human beings and how we've treated our colleagues. The way we treat research animals has changed over the decades for the good. Things like biosafety, working with dangerous organisms, or just the possibility of our human cell cultures possibly infecting us. Radiation, laser safety is important, depending on how it's regulated in your country. Here in the United States, I think it's a little over-regulated because most of our confocals are really rather safe, but we jump through those hoops anyway. And of course, chemical safety. And if you violate some of these regulations, it can result in fines or criminal prosecution, which is never fun. So in the US, looking at scientific misconduct, we have two major funding agencies, the National Science Foundation and the NIH, and they're both very concerned about misconduct, and they have similar definitions. The wording's a little different between the two, and they talk about the fact that research misconduct means fabrication, falsification, and plagiarism. And fabrication is hopefully something fairly easy to understand. It's just making things up and then pretending that they're real. Falsification, changing things that you already have to make them look like something that they aren't. And plagiarism is an interesting term. You know, one of the issues that they've discovered when they've done research is different cultures have different ideas about the appropriation of other people's work. For some people, it's actually, for some cultures, it's actually a means of extending respect to someone, quoting things in large blocks. The important thing is that you need to cite the source if you're going to use other people's work. Put it in quotes. If you need to borrow a figure, you also need to remember things like copyright, and be aware of that. And I really like this disclaimer here that it doesn't include honest error, because sometimes people make mistakes. They leave duplicate images in a publication because one was supposed to be a placeholder and they forgot. Or there are differences of opinion, and we'll come back to that. And I looked up what Sweden said online, and it looks like very similar language with some concerns about deviations from good practice and the whole issue of negligence. There are some people that just don't pay attention to the important details. So what I've seen is I've been watching this this problem for the last many years now is that most of the ethics violations fall under fabrication or falsification. There have been some cases of stolen images, plagiarized images. And it's been interesting because the whole idea of what's good scientific practice for working with digital images has taken a long time to sort of settle into what's appropriate and what isn't, because if you look at some microscopy fields, for example, in electron microscopy, it's considered okay to do certain things that are non-destructive that might be inappropriate in fluorescence microscopy. The United States National Academy of Sciences tried to come up with some guidelines, and they failed because they brought in too many people from, well, they brought in people from a lot of different image using technologies and discovered that they could not come to agreement. Imagine that, scientists that can't come to agreement. But I really try and emphasize to my users and to the people that I work with is that this clause about the differences of scientific opinion do not constitute misconduct. If you put everything out there and tell people exactly what you did to an image, they can say that you're stupid or incompetent, but they can't say that you're a cheat. And I was recently at a meeting where they had some people from the Office of Research Integrity, which is the investigation arm of the National Institutes of Health. And when I said that publicly, they sat there and nodded. So I'll take that as an endorsement.
I must say I've been quoting you on that for several years.
Good.
In a different lecture.
So how bad is the problem? Well, I understand from the course syllabus that you actually looked at this publication with Elizabeth Bick, and which is great. And it's really disconcerting that so many publications have questionable images. And that's primarily just gel images, which are a little easier for the eye to see repetitions in. But then I went to the Office of Research Integrity's case studies. These are people whose investigations were brought to the national level in the United States. They were investigated, and oftentimes they received some form of censure, inability to get grants or serve on panels. And of the cases there, about 80% of them involved falsified images. And I actually know the person from the University of Arizona who was cited in that case. They used one of my microscopes. Nice person, but apparently they crossed the line. And then I went to the Retraction Watch database, and I don't know if you've mentioned Retraction Watch. It was started many years ago. Two editors from or different journals, I forget which, who also have sort of a journalistic bent, decided to start paying attention to how retractions happen in the literature. And it's a fascinating blog to read. It takes forever to actually retract things that have obvious flaws, let alone things that might be mildly flawed. But they started this database of retractions and they labeled things. So I did a query based on, you can see the query terms down at the bottom, based on things related to images, and found that about 14% of them, of these retractions, had issues with their images. And then if you've been seen anything in the news, and I realize this is probably a little bit more US centric than it is international, but the Dana-Farber Cancer Institute is associated with Harvard University. So not just some small place in a corner somewhere in the world, a big name university. And there have been more than, I think it's like 30 or 40 images now, that of 30 or 40 publications that are in question and being reviewed. And the Times said that the images had been stretched or obscured or spliced together in a way that seemed like people were trying to mislead readers. That's, you know, as bad as it is that things happened at the University of Arizona or at a big place like that, it's unfortunately happening in a lot of places. So looking at the great lectures that you've had, and I wished I've had even more time to dive into them. I wanted to kind of review a couple of key points before we go into our small group conversation. So we're going to talk about optics, detectors, and some key point images. And I don't know if you've ever looked at the Nikon Small World contest or some of the other image contests that are held around there. People take amazing images. And, you know, it's interesting to think of that when you look at like regular photography contests, the people that typically win them have better gear than most of us do. They spend thousands of dollars on their cameras and their lenses and the computers that they use to work on their images. But for the most part, the people that win microscopy contests are actually using some of the same instruments that we have available to us. So the question becomes, well, what's the difference between their images and my images? And having worked in the core facility space for a long time now, I would argue that people that do careful sample preparation, that understand the physics of how the optics works with the sample, it's the biggest differentiator between really terrible images and really beautiful ones. And I see Sylvie nodding her head. Because it's a never-ending refrain trying to teach people that the optics of how the light interacts with the sample is a hugely important part of getting high-quality images. And things like making sure that you don't have background if that can be avoided, and that your antibody is actually labeling the thing that you think it's labeling. So let's talk about detectors. You know, this is where the analog signal in a microscope is converted to digital. And the choices made here are kind of important. You know, when we're talking about images, I know you've talked about bit depth. You know, what happens if we increase the bit depth? Maybe we have a 12 or 14-bit detector and now it goes to 16 because the files don't like those steps in between 8 and 16? Or what happens if we say, I take a 16-bit image and go, oh, I'll make it an 8-bit image? Those kind of things. Sampling. What happens if we undersample? Is there anything bad with oversampling? I can tell you with temporal sampling, many years ago with a confocal microscope, we tried to do calcium imaging on beating heart cells. And it wasn't until about a year or two later that I realized that the reason we could never get the normal cells to have a regular heartbeat is because there's a period of time between when that scan goes from the bottom corner back up to the top, missing it. And there's actually a thing called the wagon wheel effect that was discovered when they were making movies back in the early 1900s, that at certain speeds, the wheels of a wagon look like they were going backwards if you watched a Western movie, or you'll see it in some car commercials. So that temporal sampling can be an issue as well. Noise. You know, you can take a pretty noisy image and get good data from it if you're doing live cell imaging and you don't want to fry your sample. But if you are trying to measure things or get a different kind of data out of it, you may need to have less noise in your image. Is your acquisition tool capable of doing that? You know, if your signal is low, you're going to have noise. It's there. Saturation. Over in the cancer center that we have here, there's some lovely images in one of the lobbies that were taken by a pathologist who happens to be a friend of mine. And I would tell people as I walked past them, so these images are beautiful, but they're art. They're not data, because they were all very heavily saturated. And the issues with raising the black level. And I don't know, Sylvia, if you've talked a lot about histograms, but it's a good idea to keep an eye on the histogram and see what it's telling you about what's going on in your image. That's a really useful tool. And it can also be a useful tool if you're looking at an image to find out, you can sometimes see some telltale signs that there has been some manipulation in the image just using the histogram. And with digital images, I emphasize this often to people that it really is data. People tend to think of it as a picture. And because, we're on Instagram and we do filters and all that stuff, it's just a picture. We want to make it look better. But as the dictionary would say, it's information in a digital form that can be transmitted and it can be processed. And it's a numerical sampling of that analog information, which means that it has some weaknesses and it has multiple dimensions. You guys just had a talk with Peter from the open microscopy environment. And one of the things I'm learning, because we're rolling out an OME server here, is just the number of dimensions that data can have. And I often show people this image. This is a very contrasty image with a crow, I believe, or a raven. And if you look at the array of numbers that represents the pixel values in this image, you can actually see the outline of the bird, because black values in this image are low numbers and the white values are 3-digit numbers. But it's just to point out that it is actually a numerical information. Now, images come in a container, a file format. And if you've discovered everybody has their own solution to the problem. Thank God for bioformats that we have a way to translate this multiple file formats in a way that allows us to work with them. But and more importantly is that within that file format, there is metadata, data about the filters, the lenses, the microscope itself, the conditions of your laser power, all those things. That's important information for reproducibility. And exporting to a standard TIFF, because you don't want to deal with that format, can often lead to the loss of metadata. So we go from these very specialized formats to something more easily worked with on an individual computer, but we end up sending the metadata somewhere else. If you're careful, sometimes you can keep it, but then it's hard to keep it with the image. So be aware of that. And, underneath everything, when you're working with images, because they are a mathematical grid of numbers, sometimes a 3-dimensional stack of grids of numbers, any functions that we apply to an image are mathematical. And it's really very difficult to tell after the fact what's been done to an image. You have hints and tells that you can see where there might have been something that happened, but I've actually been asked to help our research integrity officer look at images, and sometimes it's like, yeah, it looks like something was done here, but I'm not sure I can tell what. And some common image manipulations are considered benign. They're not considered bad, and others are significant enough that they do need to be reported. And I often find users that work with software that they don't know what's inside the space between what they started with and what they ended up with. And they really don't appreciate why or what is happening when they're processing the images. So that was kind of a blur, but we're going to break up into our discussion groups now. And there's a list of questions. And I want you to save a little bit of time to work on the last question, because we're going to talk about that as a group when you come back out of it. And as you work through these, also think about things that you might do differently within your own work and your own lab. So Sylvie, I'll let you break people out or do what you need to do.
Yes, thank you, Doug. And I totally zapped the fact that we cannot have breakout rooms in this context. So this is why we have two Zooms. So what I'm going to do is that I'm going to send you the link to the other one, and I think we will. I will send you by email, if that's okay.
Okay.
The link to the other one. We're just going to. You're going to do some Zoom hopping, just like we do, and. So you okay, now I'm sending it to you, and you do not need any password or anything. You just join in.
Okay.
So we see you in the other room now.
Okay.
Ciao.